So first of all, thank you to the organizers for the invitation to join. This has been a wonderful conference over the last several years since its inception. Brings together a lot of great individuals where we can collaboratively amongst the pharma partners and even with the academics growing recently to think about how do we actually move dermatology forward as a whole. It's a very exciting time. We're starting to see biologics coming forward in the spaces of atopic dermatitis, psoriasis. They've been there for a while, and we're starting to think about how do we go through and treat individuals with these dermatological disorders a bit more completely. So just to start off, I want to give a bit of a background. So very interestingly, there's more than 3,000 skin diseases have been identified and described. This is an absolutely insurmountable amount of diseases to try and characterize, and trying to think through, you know, the physician. So we just heard Lotus talk about her desire to be a physician, and thinking about going through medical school and trying to remember not only just the standard diseases and the categories associated with the diseases, but then thinking about the actual 3,000 names of these diseases gets to be a bit much. In recent years, there's been about 85 million Americans that were actually seen by physicians for skin disease. And this is the largest reason that individuals actually have scheduled healthcare visits. So about 27% of the United States population has actually gone to a physician recently for a skin-related disorder. In 2013, realizing this is several years old and they're actually updating these numbers now, the annual cost for skin disease was related to $75 billion. So interestingly, obviously we all are dealing with challenging times in the midst of COVID-19, and part of that's ushered in a little bit of a paradigm shift within dermatology. Previously, a lot of dermatology relied on inpatient visits, the visual assessment of a particular lesion or an actual skin disorder, taking a skin sample, and then relying on dermatopathology reports to confirm or refute whatever the initial diagnosis was. What we're starting to see with the emergence of telemedicine is that there's this combination of both inpatient and telederm approaches. There's a increased number of patient images that are being collected from through apps and through related materials. And there's an increase in the amount of non-invasive skin sampling that's been going on as well, and leveraging that information to come up with skin genomics to appropriately diagnose, or in some cases as a companion diagnostic for potential therapies. So over the next several minutes, we're going to talk a little bit more about non-invasive skin sampling, and that's kind of the focus, is thinking about how do we go forward into this new paradigm and leverage as much information as possible. So I want to give a brief history about where non-invasive skin sampling actually started. And you've heard a couple of different individuals reference kind of the length of time that dermatology has been around, how we've done skin assessments in the last 25 to 30 years, and even before that. So very simplistically, non-invasive skin sampling was initially used to evaluate the drug penetration or skin toxicity induced by a particular compound. Using desquam discs a long time ago, they were able to go through and look at what layer of skin was penetrated, typically through a topical application. There were applications of this that modified, and in the early 2000s, one of the first publications came through looking at changes, and this was actually done at National Jewish, looking at lipidomic changes in atopic dermatitis patients. It was further evaluated using transcriptomic, so there was comparisons, because one of the big questions that comes out when you're sampling the stratum corneum of individuals with lesions is whether or not that's representative of what's going on within a full thickness biopsy. And so initial publications confirmed using transcriptomics that non-invasive tape strips actually gave very similar, about 99% overlap of skin inflammation between the actual tape strips and the biopsies. As you can see, as we move more and more to the right, there's more and more information that's actually coming out with the use of non-invasive skin sampling within dermatological disorders. At DermTech in 2015, we commercialized a melanoma detection assay called the Pigmented Lesion assay, and we'll discuss that in a little bit further as well. So one of the technologies that we've actually come up with over the last several years, and it's really coming into prominence, is what we like to determine, or like to call, the DermTech Start Sticker. You can see on the right side of your screen, this is actually one of the smart stickers. The little indentations that you can see on there is a skin sample that was collected with one of the stickers. The opportunity with the smart sticker is that it samples the entire lesion. So it can actually, rather than taking a 2mm biopsy punch or a 4mm biopsy punch, you can actually sample the entire lesion. The other parts of this is that it can serve as an early diagnostic approach. So combining not only the non-invasive sampling along with longitudinal analysis with either therapeutics or with a diagnostic platform allows you to start understanding a little bit more holistically what's going on within the patient. Leveraging this, you can come up with precision and personalized dermatology and potentially companion diagnostic approaches. The rationale behind this is that in many diseases, what we start to see in quote-unquote non-lesional skin is that there's genomic changes before there's actually a clinical manifestation to be observed. Just to provide a little bit more context and granularity to this, as we start to think about the skin, so focusing a little bit more on the left side of your screen, you can see a cross-section of the epidermis. So on the basal layers, which is the bottom left of the actual figure, and then as there's migration and differentiation of the keratinocytes as they move towards the outer layers and become the stratum corneum. As part of this, the RNA migrates, the DNA mutations actually migrate as well and can be detected in the upper layers of those keratinocytes. So one of the advantages that we found is that, again, in addition to sampling the entire lesion, we're able to go through and look objectively at the genomic information without the need for invasive biopsies that require not only anesthesia, but also require some time for the individuals to heal as well. This allows us to go through and in a longitudinal fashion, assess whether or not there are changes in the skin that would predict or could be used as a diagnostic for those individuals suffering from disorders. There's been several comparisons over the last 10 years trying to figure out what the difference is really compared back to the DSWAM discs. And so one of the things that we've actually done a head-to-head comparison looking specifically at the advantages of our technology compared to what's previously been used. So the smart stickers collect about 1.5 milligrams of stratum corneum tissue. And this actually captures the signatures from the epidermal keratinocytes, T cells, dendritic cells, and melanocytes. So we're able to see signatures associated with each one of those. The other advantage to this actual smart sticker is that unlike many methods which require immediate freezing and a cold chain process until there's a arduous process of isolation, our technology allows us to actually go through and collect the sticker at room temperature and ship it within 10 days at room temperature to our site in La Jolla, where it can then be processed in a CLIA-certified, CAP-accredited, New York licensed commercial laboratory. One of the other comments that's been made in recent years is that when working with other technologies, there's a challenge in the number of samples that can actually be processed at a time. And through the number of years that DermTech has been in existence, they've perfected this process to the point where we can analyze over 10,000 samples per month and currently do so with our melanoma assays. So as I mentioned, we do have a pigmented lesion assay that's gone forward. And so I'd like to spend a little bit of time just talking about precision dermatology, ruling out melanoma and non-melanoma skin cancer. So skin cancer is one of the most common cancers in the United States. Approximately 9,500 patients are identified with skin cancer in the United States every single day. Even more so, about 200,000 people within the United States have either in situ or invasive melanoma, and this accounts for approximately 7,000 deaths in the United States alone each year. In addition to melanoma, non-melanoma skin cancers, which include basal cell and squamous cell carcinoma, occur based on sun-exposed skin areas and account for about 15,000 deaths in the United States. Unfortunately, the current diagnosis for basal cell and squamous cell and even melanoma require an in-depth visual assessment of the area, followed potentially by a surgical skin biopsy for histopathologic review. As you can see, this is a bit of a challenge in the midst of a COVID environment where many physicians are still struggling to get their offices and their practices open. And therefore, there's an opportunity to start leveraging a non-invasive approach that can potentially be done at home. There's several papers that have gone through and looked at the comparison between a standard pigmented lesion and actual melanoma. And so on the left side of your screen, you'll see what is a not skin cancer in the upper left. That's really just a pigmented lesion or a mole. And in the upper right of that part is a skin cancer, so a melanoma. When looking at it with the naked eye, it's very difficult to see some of those differentiating factors. And when you look histopathologically at the actual samples as well, it's very difficult to distinguish to the untrained eye that there's significant differences between those two slides. Dr. Connick and his group additionally found that the delay in the appropriate diagnosis of individuals with melanoma actually increases their potential of mortality associated with it as it gets into metastasis and progresses throughout the rest of the body. So therefore, trying to detect melanoma as early as possible is a benefit. Furthermore to this, there was a study that was done a couple of years ago where they asked 187 pathologists and dermatopathologists from several United States states to evaluate several skin biopsy cases of melanocytic lesions. What I really want to bring to your attention in this is that 42% of these cases were underdiagnosed. Each site had actually gone through and used their own criteria, and then this was compared back to a consensus reference by a panel of dermatologists. And again, just to point it out, that 42 of these cases were underdiagnosed, indicating that several of these individuals may not have been diagnosed properly and therefore progressing on within their disease. So in 2015, as I mentioned, DermTech actually launched the Pigmented Lesion Assay. This focuses on the evaluation of LINC and PRAME. These are two genes that have been identified to be associated with melanoma. LINC itself is detected in 84% of histologically confirmed melanomas. And this was a novel biomarker that was actually identified using whole transcriptomic analysis of lesional skin from melanoma patients. PRAME is another gene that was actually identified. It stands for preferentially expressed antigen in melanoma. It's expressed by itself in 83% of histologically confirmed melanomas as well. The additional advantage to PRAME is that this gene itself has been confirmed and validated by other competitors, including Myriad and Castle, who also have their own melanoma diagnostics. Combining the two genes themselves, you get 93% of histopathologically confirmed melanomas express either LINC or PRAME, suggesting a high degree of confidence in using these two genes to detect the differences. Most recently, we've actually identified that the addition of a promoter mutation in TERT, which is detected in 73% of the melanomas, can be added onto this, thereby increasing our overall sensitivity, which I'll show you in a second. So as I mentioned, the combination of the TERT promoter DNA mutation with the actual analysis of LINC and PRAME increases the overall sensitivity of our PLA assay from 91 to 97%. Combined, and when you look at the rest of the community-acquired data, real-world evidence, our negative predicted value associated with this particular assay is 99%, indicating that we're capable of using a non-invasive methodology to detect patients with melanoma and rule out potentially biopsies for those individuals who just have a pigmented lesion and don't actually have progressive melanoma. One of the other areas where this becomes really interesting, as I mentioned previously, there's a large number of individuals in the United States that have basal cell carcinoma or squamous cell carcinoma, and approximately 5.4 billion of these individuals are identified each year. 8 out of 10 are actually diagnosed with basal cell, and again, this leads to about 15,000 deaths in the United States alone due to BCC or SCC. There's a known risk in these individuals with exposure to UV light or to sun exposure, and you get extensive damage that can come from this. When you look at the pictures that are there in front of you on the right side of your screen, there are different stages that are associated with these different disorders as well. So basal cell, you have the red patches, pink growths, the bumps, and the sores. And you see some very similar connections that are available associated with squamous cell carcinoma as well. And therefore, it becomes very interesting to think about how do we go through and start detecting basal cell or squamous cell from other lesions. So one of the concerns that we have is looking at ruling out non-melanoma skin cancer. So it's particularly within this assay, we identified and requested suspicious non-melanoma skin lesions from patients. And several sites across the United States delivered. They delivered 182 samples that went through histopathological review for confirmation or reclassification. Within this system, 124 were confirmed to be either basal cell or squamous cell, and 58 or 32% of those suspicious lesions were actually misdiagnosed as something else. We further evaluated using the SMART sticker, isolated the RNA from these, and then actually ran whole transcriptomic RNA sequencing. The heat map that's on the right side of your screen will suggest that there are subtle differences between some of the non-cancer and the non-melanoma skin cancers. When you dive into it a little bit more intently, in the upper right graph actually depicts one of the genes that's there. The bar indicates the median level for each of those genes. And so when you do the combination of basal cell and squamous cell and look at the separation, there's a statistically higher level of gene expression of this particular gene in the non-melanoma skin cancer compared to the non-cancer. When you look at the area under the curve or just run the ROC analysis, you can see the sensitivity and specificity, which gets you to about a 75% sensitivity in distinguishing between the non-melanoma and non-cancer. So this gives us a great idea of thinking to move forward. How do we put this in the hands of dermatologists, even primary care physicians, where there's an inability to potentially go to the dermatologist and get a biopsy to cut out? And this actually rules out the potential need for a biopsy as we go through and fine-tune this process even further. So again, just to reiterate a little bit, when we start to think about what can actually happen with non-invasive skin sampling, this enables us to enhance precision medicine in early skin detection. So by looking at the entire lesion rather than pulling a sample of a lesion, we're able to go through and assess the genomics associated with a particular disease. And as we've mentioned several times so far, there's an opportunity to see the genomic changes within the skin prior to seeing any sort of changes histopathologically or even from the visual assessments within the actual patients. One of the things that I would like to point out in this is that we're also highly accurate. So looking at the utilization of this in the detection of melanoma, it reduces the possibility of missing melanoma to less than 1%, which is great. So as you think about early detection within the case of melanoma and trying to intervene early in these patients to prevent the increased mortality, a non-invasive methodology allows you to go through and sample multiple areas on a patient rather than having to look at how many biopsies you can collect. So moving away from skin cancer and thinking about, we've already heard about several inflammatory skin diseases in this conference already, and we're going to hear more as we go through the next 2 days. There's a great opportunity for precision and personalized medicine within this space as well. One of the things that always challenges us when we think about translational approaches within clinical medicine is that the current approach is typically a one-size-fits-all. It's an ideal that we can actually go through and every patient will respond to a particular therapeutic in the same manner. This is kind of the trial and error approach. So the patient gets diagnosed with a disease. As they go through the actual disease, they look to see whether or not there's a particular response. If there's not a response, they switch over to a different drug until they ideally come to a place to get the correct drug that will ultimately lead to some therapeutic resolution. Oops. So thinking about this a little bit further, I come back to psoriasis. So psoriasis affects approximately 7.5 million people in the United States alone. It's characterized by epidermal thickening, acanthosis, so increased keratinocyte proliferation. You get raised inflamed lesions covering with a white scale. Psoriasis vulgaris is the most dominant, which has been most of the studies that have been conducted in this space so far. Lesional psoriatic skin is primarily characterized by not only a Th1, but primarily a Th17 mediated inflammation. One of the interesting pieces that's associated with psoriasis is that greater than 50% of the moderate to severe psoriasis patients do not achieve clear or almost clear with an anti-TNF targeting therapeutic, which typically ends up being the first stage in the formulary for most individuals. And additionally, you see IL-17 and IL-23 targeting biologics, which provide about 90% improvement in skin severity for most patients as well. So the question is, could you use non-invasive methodologies to start distinguishing patients or identify subsets of those individuals? Using the Smart Sticker platform, we looked at the comparison between healthy controls with no skin disease, or the normals, and looked at lesional and non-lesional skin from active psoriasis. The left side of the screen actually depicts the total RNA yield in picograms collected from both non-lesional skin and also from the psoriatic lesional skin. For those who can't see, there's roughly 35 to 40 nanograms of material that's actually collected in RNA from 4 tape strips that are collected with this sample platform. We then ran whole transcriptomic analysis and then further refined that to a heat map of 13 genes with known roles in psoriasis pathogenesis. This includes IL-17, 22, 26, IL-17F, IL-6, and IL-23, excuse me. You can see on the right side of your screen, there's a distinguishment between the normal NML, that's the healthy tissue, The non-lesional, where there's some degree of an overlap with the normal, but you can start to see some changes within the inflammation. But then as you look to the psoriatic lesions, you can start to really identify the significant differences in gene expression compared back to both the normal and to the non-lesional. One of the interesting factors that allows you to do this is, and we've seen this most recently in literature where they're starting to compare patients and identifying subsets based on gene expression within the skin, is it prompted us to start evaluating whether or not you could identify different subsets of patients within just even this small cohort. And so we specifically graphed out IL-17A, 26, 22, TNF-alpha, and IL-6 to see whether or not there were differences specifically within those pathways. As you can see for TNF-alpha and IL-6, there's a significant overlap between the potential two different psoriatic subgroups. What was very interesting is that we went through and initially stratified these patients based on their IL-17A gene expression, and you can see a relatively nice clear separation there. And if you really wanted to overpower or overinterpret the data, you could start separating them out. When we take those same patients and look at their corresponding IL-26, IL-22 levels, they're also elevated in those patients compared back to the other group, suggesting that larger studies could be used in identifying potential subtypes of patients that may or may not respond to not only TNF-alpha, but could predominantly identify those individuals that would benefit more from an IL-17 or an IL-23 targeted approach. The last disease I want to talk about a little bit today is atopic dermatitis. This is a disease that's been near and dear to my heart. I've worked in this space for over 20 years and have been encouraged by seeing the number of therapeutics that are venturing into this space. It affects approximately 10% of adults worldwide and up to 25% of children. One of the unique features that's associated with this disease, which you've already heard from Lotus before, is the intense itching or the pruritus that's associated with these patients. It's often determined to be the itch that rashes. As patients go through, the rash that forms leads to pruritus. The scratching to try and address it also leads to barrier disruption, invasion of allergens and staphylococcal antigens, which then perpetuate the cycle. One of the unique features in this is that about 50% of mild to moderate patients with atopic dermatitis are not adequately treated with current topical therapies. So when we've looked back through to see topical corticosteroids or calcineurin inhibitors, roughly about 50% of patients or less actually get to that IGA of 0 or 1, clear or almost clear. When we start to advance this into the moderate to severe population and we think about the emerging biologics, first with dupilumab, also coming forward with tralokinumab, lebrikizumab, and some others as well, we start looking at the potential efficacy of those individuals or those individual therapies. And we see that about 60% of moderate to severe patients do not achieve clear, almost clear with the current biologics targeting the Th2 axis. This itself is suggesting that there's more heterogeneity amongst atopic dermatitis than we ever observed with psoriasis previously, and suggests the difficulties in trying to think about a one-size-fits-all approach within this particular population. So at DermTech, we're moving forward into this space as well, but I want to give a brief background as to where non-invasive skin sampling has actually been for atopic dermatitis over the last 10 to 15 years. As I mentioned earlier on in the talk, one of the first studies that was actually done in atopic dermatitis actually used liquid chromatography and mass spectrometry to go through and characterize the proteomic differences between acute and chronic lesions in atopic dermatitis patients. Additionally, non-invasive skin sampling confirmed that similar epithelial profiles were there between the skin biopsies and actual tape stripping, suggesting that the tape strips themselves could be used in lieu of a clinical biopsy to be able to go through and assess whether or not changes were happening within the patients. Most recently, there's a food allergic phenotype that was also identified, specifically looking at epithelial signatures within the stratum corneum and being able to differentiate those individuals that were food allergic from non-food allergic. You can see through the rest, and ultimately one of the interesting things that's come out as well recently was the demonstration that IL-36 gamma could distinguish atopic dermatitis from psoriasis. And so if you look in the bottom right part of your screen, the figure A, you can see the elevation in psoriasis, that's the blue circles to the right. And when you look at the, again, using a sensitivity analysis, you'll compare the actual IL-36 gamma levels within atopic dermatitis to psoriasis. It's capable of separating by 100% and differentiating between atopic dermatitis and psoriasis. So in the In those instances where we're having some difficulties truly characterizing those individuals that are atopic versus some of the other lesional skin disorders, there are opportunities to think about inflammatory signals that could be used in the differentiation. So as I mentioned, the smart sticker is starting to be used more and more within the space of dermatology, particularly within a space that virtual clinical trials are moving forward. And so there's less of a opportunity to collect skin biopsies and a reliance on images from patients and trying to engage with them through teledermatology. If you start at the bottom and you work your way up, the company itself has actually expanded from working initially in atopic dermatitis and vitiligo to doing additional skin cancers. We've also done clinical studies with psoriasis lupus, and even most recently, we're exploring cutaneous GVHD, alopecia areata, and other complementary diagnostic approaches. So indicating that there's a significant breadth to the opportunity of using the smart sticker. Again, just to reiterate a little bit more about what the opportunity is, by collecting genomic signatures within the skin of these patients, you're able to go through and stratify and select patients based on skin gene expression and protein profiles. So one of the things that I would be remiss if I did not include in this is that in addition to RNA and DNA, the smart sticker is capable of collecting the microbiome and also capable of collecting protein to be able to go through and do those assessments. And we'll hopefully be able to report on that in the coming year. There's the opportunity to identify biomarkers specific to the disease or disease subsets. So we've already shown that we're capable of distinguishing melanoma from non-melanoma, specifically with the Pigmented lesion assay. And we have additional products that are moving forward to enable the detection of basal cell and squamous cell carcinomas. The other opportunity where we have several clinical trials ongoing with pharma partners is to be able to utilize this technology in tracking and predicting drug responses as well to particular therapeutics and these indications. So just in quick summary, wanted to provide a quick overview of DermTech as a whole. So we do have a CLIA-approved laboratory in La Jolla for the processing and commercial analysis of the smart sticker. This is throughout the entire United States. We've run clinical trials also with pharma partners in Europe as well. We hold broad patents for the isolation of RNA, DNA, and protein from non-invasive skin. We do have an approved diagnostic product for the identification of melanoma, and we have a growing pipeline for the diagnosis of non-melanoma skin cancers and additional inflammatory diseases. So with that, I'd like to thank everyone for their attention.